Hello friends, I am Sujata Bhargav and I am the paper coordinator for this course on plant development. The course deals with the development of a plant during various phases of its life cycle. We shall start with the zygote, which represents the start of the sporophytic phase of a plant. In subsequent modules, we shall see how the zygote gives rise to the embryo, the seed, and how the seed germinates to form the plant. Further, we shall see how the vegetative plant shows a transition to the reproductive phase, which includes flowering, gamete formation, and the union of male and female gamete to form a zygote once again. Most of the information will be based on the model plant Arabidopsis, which is amenable for genetic and molecular studies. Let us start then. In this module, we shall study the development of a zygote to give rise to an embryo in which the pattern of growth and morphogenesis of the vegetative plant is laid out. The learning objectives of this module are early zygotic divisions, morphogenesis and patterning, and how this process of embryogenesis is regulated. The zygote which is formed first shows elongation along the apical basal axis. Then it undergoes an asymmetric division to form a smaller apical cell and a larger basal cell. The apical cell forms the embryo proper and the basal cell forms the vegetative tissue of suspensor which nourishes the growing embryo. In the apical cell, two divisions occur in the longitudinal plane to form a four-celled embryo. Then one transverse division forms the eight-celled embryo. The lower cell, the basal cell, it undergoes transverse divisions to form a row of cells of the suspensor. Then there is one periclinal division taking place in the upper mass of cells to form an outer protoderm and an inner mass of ground tissue cells. So now the embryo becomes a 16-celled embryo. The uppermost suspensor cell is called the hypophysis, and this also contributes to embryo patterning. At this stage, we get what is called as a globular embryo. As the number of cells in the growing embryo increases, the future organs are specified in local regions. This process is called pattern formation. The protoderm cells continue dividing only in the anticlinal plane and they form the epidermis which covers the entire plant. Below the protoderm are the ground tissue cells which later specify the cortex and the pith. In the middle of the globular embryo are four procambial initials which will give rise to the vascular tissues of the plant. The hypophysis undergoes a horizontal division to form two cells which will give rise to the root apical meristem and the root cap in the later stages. Further patterning gives rise to the heart-shaped embryo where the shoot and root apical meristems get organized and they are separated by a central region called as the hypocotyle radical axis. Two cotyledons also emerge on either side of the shoot apical meristem. So in the heart-shaped embryo stage, most of the parts of the vegetative plant are specified and now the embryo has completed its pattern formation. Let us now study the more detailed organization of the shoot apical meristem. The shoot apical meristem or SAM appears as a distinct dome-shaped structure between the cotyledons in dicots and it appears laterally at the base of the cotyledon in monocots. The organization of the SAM, however, is similar in both dicots and monocots. The SAM shows an organization which is called as the tunica corpus organization and it consists of three layers: the outermost L1 layer, which will specify the future epidermis, and the L2 and L3 layers, which specify the ground tissue. Below these is seen a central zone, which consists of stem cells, and in the core of the central zone is an organizing center where the cells do not show very rapid divisions. Outer to this central zone lies the peripheral zone, and the central zone cells divide to form cells, rapidly divide to form cells of the peripheral zones. Below the central zone is the rib zone, and the rib zone cells also are formed from the central zone cells, and they show rapid divisions. The peripheral zone cells give rise to the leaf primordia, whereas the rib zone cells give rise to the stem of the plant. So let us see how these primordia arise. The leaf primordia are seen in the figure on the right. They'll arise on the outer side of the central zone, that is from the peripheral zone region. And these primordia, they are formed one after the other. For example, the P0 is the youngest primordia, whereas the oldest is the P3 primordium. Now, each of these leaf primordium shows an adaxial, abaxial dorsiventrality, and that can be seen clearly in terms of differences in the type of cells present in these regions. Let us now see how the root apical meristem is organized in a developing embryo. The lower tier of the globular embryo generates a cylindrical axis that defines the hypocotyl radical region. The outermost layer is the protoderm, which distinguishes from the remaining part of the axis to form the epidermis. Within the epidermal layer lie the ground tissue precursors and the vascular stem cell initials or procambium. The ground tissue precursor undergoes a periclinal division to distinguish cortical cells on the outer side and the endodermal cells on the inner side. Whereas the vascular or procambial initials divide periclinally to distinguish a pericycle initial or pericycle cells on the outer side and the vascular initials or vascular cells on the inner side. The root apical meristem is generated by the hypophysis. The hypophysis cell forms two cells by an asymmetric division, forming an upper smaller lens-shaped apical cell that divides to form four cells, which organizes to form a quiescent center of RAM. The quiescent center is functionally similar to the organizing center that we have seen in the shoot apical meristem. The larger basal cell of the hypophysis forms meristematic cells that generate the basal root cap or columella as it is called. The quiescent center cells induce the surrounding cells to become meristematic, and later these cells distinguish to form specific root tissues in the region proximal to the hypocotyle and the root cap in the distal region of the root. So let us see now how these root and shoot meristem organization is regulated at the molecular level. The regulatory processes that are involved in embryogenesis have been studied using mutants that show defects in early embryo formation. For example, three mutants have been shown in the figure to the left, namely the UCA mutants, the PIN mutant, and the NOM mutants. These three mutants have been studied to identify the genes which are mutated in them. The UCA genes encodes an enzyme called as which is a flavin monooxygenase, and it is an enzyme involved in auxin synthesis. To the right is seen an auxin biosynthesis pathway, a simplest pathway of auxin biosynthesis, wherein the amino acid tryptophan is converted by the tryptophan aminotransferase TAA to form the indole pyruvate acid or indole pyruvate. This indole pyruvate is acted upon by the flavin monooxygenase, which is coded by the UCA gene to form the IA or indole acetic acid. The auxin transporters are of two types. Some type of auxin transporters move auxin from within the cell to outside the cell, and these transporters are the efflux transporters called as PIN transporters. They are coded for by the PIN genes. The influx transporters are the OX1 transporters which bring IA from outside the cell to within. The figure on the right top shows how the auxin fluxes are carried out while the embryo is developing. So initially, you can see that there are pin proteins along the upper surface of the suspensor cells which are going to carry the auxin or transport the auxin towards the globular embryo. So there is an upward movement of auxin from the suspensor into the globular embryo. Subsequently, the auxin maximum shifts to the basal part of the embryo. So at the globular stage, auxin from the suspensor cell moves to the apical region, and then when it forms the shoot apical meristem, from the shoot apical meristem, the auxin again moves downwards towards the cotyledons and downwards towards the root. The auxin gradients define the apical basal embryonic axis. At the heart-shaped stage, therefore, you see various transport of various directions in which auxin is transported. Auxin, which is synthesized now in the shoot apical meristem, is transported to the cotyledon. Auxin, which moves up from the suspensor, is also transported upwards to the cotyledon. So there is a new maxima of auxin formed at the two cotyledonary poles. This maxima then leads to a downward flux of auxin towards the base of the embryo. All this transport of auxins is carried out by the PIN efflux carriers. Besides auxin synthesis and transport, perception of this hormone is important for initiating an auxin response in the growing embryo. Within a cell, the auxin response genes are regulated through an auxin signaling mechanisms. Some of the genes which are involved in signaling of auxins are shown in the table. They include auxin response factors, which are positive regulators of auxin response genes. Then you have the auxin IA or AUX-IA factors, which are negative regulators of auxin response genes. You have an auxin receptor called as the transport inhibitor response 1 or the TIR1. You have examples of the auxin response factors, which are monopteros, and an example of the AUX-IA response factors, which is Boden loss. And there is another signaling gene called as the topless or TPL, which bind to the Boden loss factor to repress MP. We shall see more details about how this auxin signaling takes place. And this is shown to the right. In the absence of IA, the auxin response genes show the presence of auxin response factors attached to the promoter regions, but these auxin response factors cannot carry out their activation of the auxin response genes because they are inhibited by the repressors of auxin responses, namely the OX-IA factors. The topless is further going to repress the activity of the ARF genes. The auxin receptor TIR1 lies vacant in the absence of IA, and hence the auxin response genes are in a default shut position. In the presence of auxin, the TIR binds to auxin, and this TIR is now activated. It will bind to the auxin IA repressors and remove them from the auxin response factors. Once these repressors are removed, the auxin response factors or the ARFs are now able to carry out positive regulation of the auxin response genes. The TIR bound to the auxin IA factors are marked for ubiquitination and degradation by the 26S proteasome protein degradation machinery. After having understood that auxin plays an important role in the early embryo development, let us now see some of the genes that are involved in the actual patterning of the zygote and the early embryo. The first gene which is involved is a gene which comes from the male parent, that is from the sperm cell. It is the short suspensor gene, and this gene activates a signaling program in the developing zygote and leads to a first asymmetric division of the zygote. So therefore this gene seems to link the fertilization event to the development of the embryo, and the involvement of the male parent is important for subsequent embryo development. The next gene is YODA, which is responsible for the cell fate of zygote derivatives. It is a component of the MAP kinase signaling pathway. Then there are Bushell related homeobox genes, which specify the apical and basal domain of the early embryo. Finally, there are two genes which are playing an important role in specifying the protoderm. These are transcription factors of the HD-ZIP type. First is the Arabidopsis thaliana meristem layer 1 gene, and the second is a gene which codes for a protodermal factor PDF2. On the right is shown a tentative signaling pathway involving these genes. The pollen-specific short suspensor protein is responsible for activating the YODA signaling pathway. The YODA signaling pathway leads to elongation of the zygote. Subsequently, the Wuschel-related signaling pathway operates, and this pathway then specifies the apical and basal domain of the early embryo. Then, when the globular embryo forms by the cell divisions, the two genes ATML1 and PDF2 are responsible for differentiation of the protoderm in the globular embryo. Let us now see some of the genes which specify the organization of the shoot apical meristem. There are several genes involved in specifying the shoot apical meristem. The Wuschel gene or the Wus gene, it is responsible for specifying the organizing center in SAM. There are three clavata genes, namely CLV1, 2 and 3, which form a receptor kinase signaling pathway, and it specifies stem cells and restricts the Wus expression to the organizing center. So therefore, Wus and clavata form a feedback type of a loop. Wus is expressed in the organizing center, and clavata represses the Wus expression in the region outside the organization center in the stem cell region. So therefore their domains are totally restricted away from each other. Then you have Other genes like the shoot meristem-less gene or the STM gene, which specifies the stem cells in SAM. Then you have the cup-shaped cotyledons or the Cook gene, which defines the boundaries between stem cells and the organ primordia. And finally, you have asymmetric leaves genes, which specify the founder cells for the lateral organs. So the domain in which the shoot meristem-less gene is expressed is in the region above the organizing center, in the region where the stem cells undergoing rapid divisions in the SAM are present. The cup-shaped cotyledon genes are also expressed in this region initially, but later as the embryo moves towards the heart-shaped stage, cup-shaped cotyledon genes, they define the boundary between the stem cells and the organ primordia. So you can see that the shoot meristem-less gets restricted to the central region, which is which represents the SAM, and the cup-shaped cotyledon genes are expressed on the periphery where STM gene is not expressed. The asymmetric leaves genes are expressed in the region where the cotyledons emerge, and they establish the founder cells for lateral organs. So you see that the shoot meristem-less genes define the meristematic region in the SAM. The cup-shaped cotyledon genes define the boundaries between the organs and the shoot apical meristem. And the asymmetric leaf genes which define the organ identity. Like the shoot apical meristem, a number of genes are involved in specifying the root apical meristem. These genes are expressed very early, right from the formation of the globular embryo. The plethora genes are the first group of genes which are expressed in the globular embryo, and they are auxin-responsive genes which are expressed in the procambial and the cortical region, the ground tissue region of the globular embryo. The short root gene is also expressed in the globular region, but its expression is restricted to the procambial cells. The scarecrow gene in the globular embryo is seen to be expressed only in the hypophysis cell. When the development proceeds to the heart-shaped stage, the short root gene from the vascular region, the short root protein moves towards the ground tissue region and leads to the induction of the scarecrow gene in the innermost layer of the cortical cells to form the endodermis. So you have the scarecrow gene which specifies the endodermis, the innermost layer of the cortical cells, and the plethora and short root genes which specify the vascular cylinder towards the center of the heart-shaped embryo. Now for vascular development, a local increase in the auxin concentration is necessary for specifying the vascular stem cells. The identity of the vascular uh, cells is attributed to the auxin response factor monopteros, which induces the expression of ATHB8 or the Arabidopsis thaliana homeobox gene 8, and this gene specifies the procambial cells. In addition, there are two more genes which are activated by monopteros, namely the target of monopteros gene or the TMO5 gene and the lonesome highway gene, both of which are involved in the development of the vascular stem cells. So monopteros, which is induced by the building up of an auxin maxima, leads to the activation of genes like TMO5 or lonesome highway or ATHB8, all of which contribute to vascular development. In addition, There are some cytokinin biosynthesis and signaling genes which are also involved in specifying the vascular stem identity. So you have the lonely guy gene or the log4 gene which specifies the formation of the vascular elements, and you have the wooden leg gene which is involved in cytokinin signaling. So both the log4 and wol genes are expressed within the vascular stem cells along with the genes which are specifying which are induced by auxins, namely the TMO5, the lonesome highway, and the ATHB8 genes. Together, the auxin and cytokinin responsive genes bring about the development of the vascular cylinder. At the end of early embryogenesis, we have the heart-shaped embryo which lies within the ovule and is protected by the ovule wall layers. The ovule wall layers later form the seed coat. The embryo consists of an embryo axis with the shoot and root apical meristems at either end of the hypocotyle and the cotyledons. In addition, endosperm development starts from the fertilized central cell. We shall see the further development of this embryo in the next module. Thank you.